Okay. Thank you for the introduction. My name is Omar. I'm a graduate student from Johns Hopkins in Dr. Ben Langmead's lab. And today I'll be talking about a new data structure for document listing in pangenomes. So I'll start by talking about read classification, which is kind of a common task in many genomic workflows. So typically, if you have a DNA sample and you want to figure out what's the contents of it, you'll kind of do some sequencing. You have a set of sequencing reads. And then you will sort of grab a set of genomes from a reference database and then build an index. And typically, we have software tools that are FM index based or Kamer based. And then you go combine these two and get a set of read classifications. And notice how some of them are more specific than others. We could have some at the species level, some at the genus level. So this is kind of the overall workflow, and we'll kind of use this to kind of guide some motivation for this project. So first thing, if we look at the reference database, it's not very realistic. Nowadays, we have a lot of assemblies, we have a lot of species, so it puts a lot of pressure on indexing software to build indexes. And one of the issues that can come up is that these indexes can grow scale linearly, which can make it a little impractical for building indexes over entire genomic databases. And the second issue that can kind of occur is when you look at some of the classifications, there's been previous work that's looked at Kamer-based methods and showed that as the database gets bigger, you have less reads being classified at a specific level, at the species level of the tree. So don't worry about interpreting the plot. It's really just a way of showing that as the databases get bigger, classifications tend to get less specific, which is obviously not ideal for whatever you want to go and use those classifications for downstream. So now I'll kind of do a little background on a data structure called the R-index, which we'll talk about later in the talk. So what is the R-index? So if you're familiar with the FM index, it's essentially a pangenomic version of that, allowing you to do the similar types of queries, such as present-to-absence queries, locate queries, count queries. And what makes it sort of adaptable or optimized for pangenomes is that the size of it tends to grow sublinearly as you add in similar sequence into it. And the reason for this is that its space complexity, as the name sort of implies, it's a space complexity O of R, where R is the number of runs in the BWT of the text. And the key idea that kind of allows it to scale efficiently is that as we add in similar sequence, the number of runs in the BWT of the text tends to grow slower than the total length of the sequence. So here's a little example. We have a very repetitive string of recombs, and when you compute the BWT over it, you can see that we have a sequence of runs, and the number of runs is equal to 8, while the total length is 21. So if you run length encode that, you get a smaller representation. And if we look at some examples on E. coli genomes, we kind of see a similar effect where you can see the length of the sequence N is growing linearly, but when you look at the number of runs, it grows fast in the beginning, but then it starts to peter out. And that allows us to kind of have a more efficient representation of a pangenome. So with the R-index, if we wanted to do something multi-class classification, like taxonomic classification, how could we do it just theoretically? Well, we could do the standard backward search algorithm where you have a Burroughs-Wheeler matrix. Say you're looking for the pattern ACAT. You kind of do backward search, and you see you have this range. And then you could do a locate query for each occurrence, and then kind of track that back to what genome it came from. But the problem here is that as you have to do some work for every single occurrence in the pangenome. So as we can imagine, we're adding more assemblies. That means we're going to have to do more and more work for every single time we want to do any sort of pattern matching. So this is kind of the baseline, and we hope to kind of develop an approach that's better than this. And then the last bit of background I want to give before going into the method is I want to talk about this tool called Spumoni, which I've worked on, that's based on the R-index for doing read classification. It was initially developed for binary classification applications, and since then we kind of extended it to multi-class classification against pangenomes. But we realized it could be better, and I'll kind of explain why in a second. So right now, for what Spumoni uses as a data structure we developed called the sample document array. So essentially, if we have an example where we have 3 genomes in a database, so you can think of these, we call them technically documents 1, 2, and 3. So if we have a BWT, and we can see the runs are kind of colored differently. Basically, what Spumoni does is at the start and end of each run in the BWT, it looks at that suffix, and it just determines, based off the suffix position, what genome does it occur in, and it stores that in the document array. And that allows us to get some kind of multi-class information, you could say, to be able to classify where that read comes from in the database. So we have a read, say you have a substring P from that read and you want to classify it. So the natural question to ask is, what genomes does that string occur in? And the true answer might be that it occurs in genomes 1 and 3. But right now, with Spumoni's kind of sample document array, we would only be able to get one of these documents. So we're not getting the full listing, but so that's kind of the main weakness right now. And that's an area that we want to kind of improve on with this work that I'll talk about today. And before moving on to the next slide, let me just define some terms. So D is equal to 3, so we have 3 documents. So that's what D stands for. And then another term, ndoc, refers to the number of documents that a substring occurs in. So in this case, this pattern, since I mentioned, occurs in genomes 1 and 3, we would say ndoc is equal to 2. So now getting to this work and to describe what we want to accomplish. So we want to develop an OVR data structure that can sit alongside a run-length encoded BWT and allows us to get full document listings in O of M plus ndoc time. So if I kind of parse out this statement, when we say document listings, all I really mean is I want to figure out all the genomes that a substring occurs in. And looking at the complexity, so M equals the length of the pattern, and then ndoc refers to the number of documents the pattern occurs in. So this is really kind of the optimal time you could hope for, where you have a substring, you have to process that substring, and then you want to go and figure out what documents occurs in. So this is kind of the overall goal we hope to achieve. So getting into this problem of document listing, which we sort of did an example on in the previous 2 slides ago. So document listing is sort of a well-studied problem. There's been solutions proposed in the past, and I'll just kind of quickly summarize a couple. So Muthukrishnan and Sadhkan developed kind of 2 document listing solutions. And the main thing I just wanted to highlight is that both of them have space complexities proportional to that are linear. And this is not ideal for pangenomic applications where the databases are so big at this point. It makes it a little impractical to use a data structure with that space complexity. So we wanted to sort of develop a smaller approach. So we developed a new data structure called document array profiles, which has a space complexity of R times D. So just to kind of highlight some of the contributions, since I won't be talking about all of them, we just kind of define the data structure, improve the theorems on its size. We discuss how we can just construct this data structure on pangenomes efficiently, and how we query it. So I'll be mostly focusing on the first and third one. And feel free to ask questions about the second one if you're interested. So if we could just go through a quick example to kind of see what the data structure looks like. So if you have 2 genomes, which is kind of the simplest multi-class example we could look at. So again, we'll call them document 1 and 2. So we have the Burrows-Wheeler matrix, our BWT, and then we have a DA, so the document array, which we'll kind of use just to kind of understand this example. So that's essentially telling us what document does that suffix occur in. So the suffix is the blue part. So where does that suffix start? So it's either going to be in document 1 or document 2. And then the PDA is the document array profile. So if we kind of try to understand this data structure, so the number of rows is equal to the number of runs in the text, and then the number of columns is equal to the number of documents. And I don't know if you could see that, but that's what it says. So I have a little red asterisk there because I'm actually showing you all N profiles, but in practice we would subsample this to only R profiles. Okay, so now let's try to understand what we're sort of storing in this data structure. So it's essentially a matrix of LCP values. So what's an LCP value? It stands for longest common prefix. So here's a little example with 2 suffixes. So essentially you want to kind of just capture how much of their prefixes do they share. So we could see that both have an AT in the beginning, so the LCP would be 2. And then next, we can kind of go through one of these rows and see if we can just understand intuitively where it came from. So for the row in row 4, so how did we get the entry 9, 2? So 9, since it's in the first column, we'll be referring to document 1, and we're asking the question, what's the maximum LCP between the suffix in row 4 and any suffix from document 1? So that's sort of a trivial example because if you look at row 4, you'll see that there's a 1 in the document array. So it sort of would match with itself as the maximum LCP. So the length of that blue part is 9. So it would be, that's where the 9 comes from. The second one's a little more interesting. So where does the 2 come from? So we asked the same question: what's the maximum LCP between the suffix in row 4 and any suffix that occurs in document 2? So if you look right above it in row 3, you'll see kind of just visually that's probably the most similar suffix to it. It also has an AT in the beginning, so that's the maximum LCP. So that would be 2. So that's kind of just defining the data structure and kind of understanding what the values in it represent. And then next we can kind of go into querying. So this is sort of the formal definition of what we kind of described previously with words. You don't have to worry too much about the definition. You can just think of it less formally. Basically, the maximum LCP between every suffix I and all the other documents. So if we go back to kind of the same example we had on the previous slide, now we want to query. So we want to take a string and figure out what documents does it occur in. So we have the pattern AT, and we'll query it against kind of this database. So the first thing we do is we do backward search, and visually we can just kind of quickly figure out that those 2 suffixes are the only positions where we have AT occur. And the first thing we do is we extract one of the rows of the documentary profiles data structure. So we can either extract row 3 or 4. So we can just extract row 3, for example. And then the next thing we do is we just kind of simply scan through the profile and we ask the question, what entries are greater than or equal to the length of our pattern? So in this case, our pattern is AT, so the length is 2. So we kind of just scan through and we realize, okay, 2 is greater than equal to 2, 3 is greater than equal to 2. So that means it occurs in both document 1 and document 2. So that's kind of our document listing. So you can see that we did some work proportional to the pattern, and then we did a simple scan to kind of just get the listing exactly. And the key idea here is notice that we're not doing any work proportional to the size of the backward search range. We're just doing a simple scan across one of the rows of the data structure. So kind of going into some results, here are kind of just the main experiments we'll be talking about, looking at the query time using this data structure versus using locate queries with our index, and then also discussing how this data structure, does it help us improve classification performance compared to Spumoni, which I talked about in the beginning, which kind of had that limitation of only giving you one of the values of this document listing. So for the first experiment, we wanted to kind of assess the impact of the database size and the number of documents. So these kind of 2 factors here on the query time. So we're comparing this new data structure, the documentary profiles, to the R index, which we talked about in the beginning, that uses kind of locate queries as typical FM index. So we had a 3-class, a 5-class, and an 8-class sort of scenario. And I don't know if it's readable, but you can kind of see we're just having different numbers of bacterial species included in each. And then we vary the size of the database. So we had a 30 genome, 100 genome, and a 300 genome scenario. So an example would be a 3-class, 30 genome database. So we had 10 genomes from each of the 3 classes. So the actual experiment, we're just kind of measuring the query times it takes to get those document listings that we did an example of for maximal exact matches. So just to quickly define MEMS, you can think of them as essentially substrings from your read that you can't extend to the left or right anymore and still find an exact match in the text. So essentially just substrings that we're going to be using to query our database. So what we would do is we would extract kind of 1 million of these MEMS for each class. So for a 3-class scenario, we'd query with 3 million MEMS and just compare the kind of the query time using the R index versus using this new data structure, the documentary profiles. Okay, so you can kind of look at some results. So here's just the legend. We can kind of decode this. So we have the doc array optimized. So basically the same thing. The only thing was we're using a heuristic to kind of reduce the space. But other than that, you can kind of treat them the same as your mind. All we're doing is sort of storing it in small space to kind of reduce the size of the index. And then we're comparing it to the R index. And then we'll have the different colors represent different numbers of classes in the database. So remember, the space complexity of the documentary profiles is R times D. So as we add more classes, the space sort of increases. So this panel over here, this we're looking at the 30 genome database. So you can kind of see the squares are the R index. So first off, let me explain. So the x-axis is the index size, the y-axis is the query time. So you can see the R index are the square points. You can see that they're pretty much vertical, which makes sense because it doesn't really matter how many classes you have because it doesn't have a factor of D in the space complexity. But when you look at the triangles and the circles, you can kind of see how they increase to the right because we have more and more classes. And then the second thing to notice is that the triangles and circles tend to be lower than the squares. And then as you kind of look at bigger and bigger databases, so 100 genomes, 300 genomes, you can kind of see that the squares are going up and the triangle and circles are kind of going up but mostly staying flat because as you have more and more genomes, you have more and more occurrences for every substring, so you're doing more and more work, more and more locate queries. So if we just focus on one of these examples, for example, the A-class scenario, you can kind of see the R index is scaling. It has a much steeper slope, and the dock array is a little more flat. So this gap is, like I said, is due to the fact that you have to do work proportional to every single occurrence that you have in your database for a pattern. So kind of the key conclusion to take away from the slide is that our query time is sort of independent of the number of occurrences of the pattern, since it doesn't have to do any more work if there's one occurrence or 100 occurrences. So transitioning into kind of the second experiment, so we kind of had the hypothesis that if you have more information, if you have the full document listing, we could get better classification accuracy compared to kind of the original approach with Spumoni that only gave you one document per match. So we kind of created 4 data sets of increasing difficulty, and each data set had 4 classes, so it's a little confusing. But so you can see we have these 4 data sets, very simple, kind of just like taking 4 classes of different genera or 4 classes in the same genus. And you can see how they have increasing levels of A and I, so it's going to be harder and harder to differentiate them. And then here's kind of just a little empty plot to kind of understand what we're going to look at. So we have the 4 classes, and we're looking at precision and recall. And then we're just going to be comparing the document array to Spumoni. So here's 2 of the data sets, so the different genera and same genus data sets. You can see that both of them are basically performing perfectly, which makes sense because it's sort of a relatively easy task to distinguish sequences from either different genera or the same genus based just off A and I. When you look at a more complex scenario of sort of strain level classification, you can see that the bars are fluctuating a lot more. But generally, the doc array bar, or the red bar, is always higher than using Spumoni, which makes sense because it just has more information to use. So the key takeaway here is that for highly similar sequences, we do see that using a full document listing helps to improve classification. So in terms of key takeaways, we talked about this new data structure called the document array profiles that scales well with pangenomes and allows you to get full document listings for substrings. Right now, it has a space complexity of R times D. So if we circle back to those issues we talked about in the first slide, so we talked about this problem of indexes scaling linearly for pangenomes, which is problematic due to their size. The document array kind of provides a sublinear option that could sort of sit alongside a run length encoded BWT for doing things like classification against pangenomes. And we also talked about this issue of how when you use a fixed length substring like Hamer's, there's issues of specificity decreasing as you use bigger and bigger databases. And this allows you to get listings for kind of variable length matches like MEMS, which presumably would kind of give you maybe more specificity in regions that you can afford it. And then lastly, kind of the major drawback of the data structure right now is it does have that factor of D in the space complexity. However, we do have some experiments that we didn't talk about right now, but show that we can probably compress the data structure to O of R and still kind of get good classification in taxonomic scenarios. So that's the end of my talk. Thank you to everyone in my lab. Thank you to my co-authors, and I'd like to acknowledge the funding sources. Thank you.